peoples who are joining us today in person and online, and to the traditional custodians of the lands from which our online attendees are joining. The sovereignty of this land was never ceded. It always was and always will be Aboriginal land. Today we have, as I mentioned before, a couple of guest speakers from the Queensland University of Technology. Very excited to have them join us today. They were originally invited by one of our clinical staff, and he has just arrived in the room. There you go, Rob. I'm Rick. So today we have Mr. Sam Torrens. He's a PhD candidate from the Queensland University of Technology. Prior to commencing his PhD, he studied exercise science at RMIT, graduating in 2010. He completed his undergraduate degree and gained an interest in exercise metabolism research and undertook his research honors year at ACU with Professor John Hawley. Sam then took several years away from study to travel abroad. He lived in Italy and Canada, and after 5 years, he returned to Australia and is now in his final year of his PhD, working with Professor Robert Robergs at QUT. Sam's area of research interest include exercise metabolism, biochemistry of substrate metabolism, metabolic acidosis, and metabolic disorders. And the title of his presentation is Carbohydrate Ingestion Before Exercise in McArdle Disease. Does it really work? Sam, if you want to go ahead and come up, I'll just get you organized over here. Okay, the floor is yours.
All right, thanks very much. Thanks for everyone for coming today. I'm presenting my PhD work under Professor Robert Robergs today, and it is carbohydrate ingestion before exercise in McArdle disease. Does it really work? So I'd firstly just like to acknowledge we got funding for this project, or for the 2 studies of my PhD. Through IMGSD, which is the International Association for Muscle Glycogen Storage Disease. They're a patient advocacy group, international patient advocacy group. They've been really supportive of my work, so a bit of a shout out to them. So first of all, McArdle disease is super rare. Has anyone heard of McArdle disease before in the audience? No? Okay, well, hopefully this is relatively educational for you all. So, oops, are we clicking or are we? That looks better. All right, so McArdle disease, genetic myopathy affecting skeletal muscle. It is a deficiency in the body to produce the enzyme muscle glycogen phosphorylase. So glycogen phosphorylase catalyzes the first step of glycogenolysis, where glycogen is then broken down into glucose to then proceed through glycolysis. Incredibly rare, so it's 1 in 100,000 roughly, but it could be as large as 1 in about 150,000. So small research field, small amount of participants to do research on, which is challenging, but we're doing all right in those areas. But it generally displays as physical activity intolerance. Now, physical activity intolerance in not just exercise, but activities of daily living. A lot of the time, people just think it's exercise. They can't do exercise, but it's anything from walking up a set of stairs to brushing your teeth, where they don't have access to skeletal muscle glycogen, and that becomes a real issue. And I'll get into that as to why. But generally, it presents during physical activity as increased heart rate, severely increased heart rate, increased perception of effort, premature fatigue, muscle stiffness, cramping, and what's also known as McArdle's contracture. So they get fixed contractures where their muscles completely seize, which is still being researched as to why this occurs. It's also important to note here that it's skeletal muscle glycogen that they can't access. They have full access to liver glycogen. So phosphorylase is expressed in the liver, just not skeletal muscle. So what does that actually look like? So in a non-McArdle individual, we've got the pathway for glycogenolysis here. So you have skeletal muscle glycogen, which can then be phosphorylated into glucose 1-phosphate. You can have the flux through glycolysis, which will then spin off pyruvate in the presence of oxygen. We can have those pyruvate go into mitochondria for mitochondrial respiration, or in the presence or in the lack thereof of oxygen, we produce lactate. Now, McArdle's disease, also with that as well, we would have the increase in hydrogen ion production through glycolysis, which leads to an increase in acidity or a decrease in pH. Now, if you have McArdle's disease and you don't have glycogen phosphorylase, you have the blockage in this first step. And so when individuals in McArdle's disease either start to exercise or start to produce or require higher levels of ATP, this is blocked. So what we don't see is the flux through glycolysis, which means we have decreased levels of pyruvate, decreased levels of intermediates coming from the TCA cycle, then going into mitochondrial respiration. And classic symptoms of the condition, if you can measure blood, is they don't produce increases in lactate and they don't produce increases in hydrogen ion concentration increases. So they don't produce this increase in acidity. Another classic case or feature of the disease is called the McArdle's second wind. Now, after about the first 6 to 8 minutes of exercise, individuals with McArdle's disease start to exhibit increases in exercise tolerance. So they go from really high ratings of perceived exertion, really high heart rate, and then after about 6 to 8 minutes, this starts to drop off. And the reason that this starts to drop off is because they start to get increases in substrate arrival from liver glycogen, because again, phosphorylase is expressed in the liver. So they get increases in liver glycogen delivery. And why that's important is liver glycogen or blood glucose, I guess, comes in the back door for glycolysis. It doesn't have to go through glycogenolysis. And so they can start to get an increase in the flux through glycolysis, so then they can produce more pyruvate, again, more lactate without presence of oxygen, or the TCA cycle intermediates, electron transport chain activity under these circumstances. But they also get an increase in lipolysis. So after the first 6 to 8 minutes, they get an increase in lipolysis, so free fatty acids in the blood. They can then enter the working muscle. Obviously, they don't go through glycolysis, but they undergo beta oxidation within the mitochondria to produce the TCA cycle intermediates and then onto the electron transport chain. So in the early study of McArdle's disease, what they did was it's relatively rare, relatively new in terms of its discovery. So it was only discovered in about 1951. But when they first discovered McArdle's disease, they took biopsies of individuals with McArdle's disease, put them in petri dishes, and they either put muscle or glycogen in with them, or they put in glucose 1-phosphate or glucose 6-phosphate. And what they found was that if they used the glucose 1-phosphate or the glucose 6-phosphate, they would get a flux through glycolysis, increasing pyruvate, lactate, etc. But if they put in glycogen, there was no flux. And so that's how they could work out that muscle phosphorylase was the enzyme that was causing this issue. So when they started to work out, okay, how can we manage the condition? They worked out that, okay, if we increase blood glucose levels before they start to exercise, we can prematurely induce this McArdle's second wind. So what they started with was infusion. So they had cannulas into individuals with McArdle's disease, and they would infuse them with high levels of glucose. And what they could find was that they would get increase in exercise tolerance because they were getting this flux coming from blood glucose that would rescue the energy crisis. But obviously, having a cannula in your arm constantly in a constant supply of glucose isn't really a practical measure of managing the disease. So what they decided to try after that was, okay, what if we did it with the ingestion of high levels of carbohydrate? So that's where the idea of, okay, let's induce hypoglycemia with things like soft drink, or what they used was sucrose. So the original sucrose study was done by Vissing, and again, it shows how recent the research has been done around this. This was only done in 2003. The previous cannulation studies or infusion studies were done in the 60s and 70s, but the idea of ingestion was only around 2003. And so what you can see here is that we've got exercise time on the x-axis, so 5, 10, 15 minutes, and then we've got exercising heart rate. And what is also important to point out here is the workload. So I don't know if anyone cycles here, but the constant workload that they were set at was 50 watts. Now, for most people that would sit on a bike and do 50 watts, that would feel like no resistance at all. But for individuals with McArdle's disease, you put them on a bike at 50 watts and they really, really struggle to pedal at those watts. So it shows the energy crisis and the deficit that they have when trying to perform any physical activity. Again, not just exercise. But as you can see here, when they were ingesting high levels of sucrose, so this was 75 grams, 30 to 40 minutes before exercise, it would show that they would have a far lower heart rate, and these are the black circles, compared to if they had a placebo, taste-matched placebo. So they're getting heart rates up around 160, and again, this is at 50 watts. But if they ingested the sucrose, they would have much lower heart rates, peaking at roughly 120. So that need for that second wind was induced straight away from exercise. Now, this was a step forward in practicality because now you didn't have to have cannulation. However, if you needed to wait 40 minutes before exercise or 40 minutes before activity, it wasn't really that practical. So what the Anderson group did was, well, what about if we tried 5 minutes before exercise? Because theoretically, if you were in a workplace, you could be going to lunch out with your group and someone says, oh, we're heading out for lunch in about 5 minutes. Do you want to come for a walk up the road to get lunch with us? Now, for someone with McArdle's disease, just walking up the road, a couple of 100 meters, will take them several attempts because of their low activity tolerance. So what they could do is say, okay, 5 minutes, I'll just finish this email, have their 37 grams of sucrose or 75 grams of sucrose, and then theoretically be able to walk to lunch without having stops and having exercise intolerance. So what Anderson did was a similar trial to Bissing, where they had individuals exercise at, it was 65% of their VO2 max, but again, around 45 to 50 watts, so very, very low absolute intensities, but high relative intensities for these individuals. And we can see a similar pattern. So again, we've got heart rate on the left, time on the right. And so we're looking at the squares. Here is the placebo. So we see the increase in heart rate without sucrose ingestion. And with the circles, that's with sucrose ingestion. Now, the black circles are 5 minutes before exercise, and the white circles are 5 minutes before exercise. Sorry, I've got that around the wrong way. 5 minutes before exercise is the black circles. 40 minutes before exercise is, oh my God, I've messed that up again. Sorry, you can probably see what it is. 40 minutes is the white circles, black circles is 5 minutes, and they were both taking 37 grams of sucrose. Sorry. And what they deduced from this was that in the second half of exercise, RPEs and heart rates were lower for the group that took the sucrose 5 minutes before exercise. And so because of this, this came across as a really practical method that individuals with McArdle's disease could use not only during exercise, but activities of daily living to reduce the conditions, because it looks like it's almost a magic bullet. You have this sucrose before exercise and your symptoms are gone. So as a result of the Anderson study, there were guidelines that were written for McArdle's disease. And again, this shows how new this research is. The first clinical guidelines for McArdle's disease were written in 2021, but their current dietary management advice is to ingest 37 grams of sucrose 5 to 10 minutes before exercise. Now, I've been involved a little bit with the McArdle's community, as my sister has McArdle's disease. And so growing up, I experienced my sister's exercise intolerance and how it affects her life. And going from doctor to doctor, that would continually say, have sucrose 5 minutes before exercise. And my sister has always sworn black and blue, it doesn't work, or it makes her worse. And so this was the starting point for my PhD, where I thought, okay, if it doesn't work for Sophie, is it just Sophie that this doesn't work for, or is it other people as well? And so I started interviewing individuals around Australia with the help of IMGSD, and the same thing kept on coming up, that carbohydrate doesn't work before exercise for individuals with McArdle's disease. And so as part of my PhD, my second study, we devised a survey of 27 questions around individuals with McArdle's disease exercise habits, their experience with carbohydrate before exercise. One of the main questions we asked was, do you find the current McArdle's disease management guidelines of consuming sugary drinks or foods before you're physically active relieves or minimises your McArdle's symptoms? Now, what was really interesting for us was that 40% of the individuals, of 102 individuals with McArdle's disease, said no, doesn't help. Doesn't help at all. And I actually got a lot of pushback, a few angry emails saying you shouldn't be promoting carbohydrate before exercise. This is old research, it doesn't work, we know it doesn't work. So it's quite a contentious topic. We also found that 29% of the individuals said yes, sometimes it works. So they could get it to work sometimes, not other times. And what was really interesting, only 18% said yes, this works. So you've got research showing really clear benefits of this almost magic bullet like, wow, my symptoms are gone, to when you transfer it to real world settings, okay, maybe it's not so. Now, there are multiple reasons that we feel that could be the case, some being around the guidelines not being overly detailed, and a lot of information that we were finding was in the taking of the carbohydrate, that it was either taken at the wrong time, not enough carbohydrate. We had a few small handful of individuals that were drinking sugar-free Red Bulls or sugar-free soft drinks, so not understanding the concept that you need to induce hypoglycemia before exercise for it to actually work. But what we thought was it was probably a smart thing to do to go back first and look at prior research to see, because physiologically to us, it makes sense that if you induce hypoglycemia beforehand, you have more substrate that can come in the back door with glucose-6-phosphate and provide the flux through glycolysis. So we went back and looked at the research that currently props up the current guidelines of 5 to 10 minutes before exercise. And the first thing that we noticed was that this is the group where they did 40 minutes versus 5 minutes versus a placebo. And we looked at their blood results, and their plasma glucose results sort of jumped out to us, because what these trials stated is that the individuals were overnight fasted when they came to the lab. But their overnight fasted blood glucose levels were 7 millimoles. Now, for anyone who understands blood glucose levels, an overnight fasted blood glucose level should be between 4 and 5.5. So the individuals who have done this trial are either insulin compromised or they haven't followed the overnight fasting protocols. So if you look here, that they're pre-primed with glucose before they actually even start the trial. So it hugely advantages the 5-minute trial over the 40-minute trial, because the 40-minute trial allows for digestion of the glucose to get into your bloodstream and then elicit hypoglycemia, which I'll get to in a second, but takes about 30 minutes. Whereas after 5 minutes, it would be very questionable how much of that blood glucose could have got into your blood. So we found this is a bit of a hole maybe in methodology to be looked at further. So we know physiologically that blood glucose response to carbohydrate loads looks very much like this. And this is a standard oral glucose tolerance test where if you look just from the 0 point onwards, that's when these individuals, these aren't McArdle's disease, these are just general non-McArdle's, with different glucose loads of 25 grams, 50 grams, and 75 grams. So anyone who's been around someone with diabetes, when you have an oral glucose tolerance test, it's the same thing that they're essentially doing here. Give them a glucose load, and you can see after 30 minutes, that's when it peaks. So theoretically, that's when individuals with McArdle's disease should start exercise, because that's when they've got the most substrate available. But if you see the little orange arrow there, that is 5 minutes after ingestion, and you can see there's been no real change. So physiologically, it doesn't make sense that 5 minutes before exercise would be a superior method. Now, in knowing this, with my third study, we are testing the Anderson and Vissing trials against each other, but as a precondition to our entry into the trials, we're doing glucose tolerance tests with our participants. And what we've found so far, we've completed 4 individuals so far, And when they do glucose tolerance tests with 37 grams of sucrose, we can see the exact same pattern. So after 5 minutes, so you've got time here on the x-axis and on the y is blood glucose levels, that they're coming in overnight fasted. And again, they have blood glucose levels within normal ranges, so roughly 4.5 to 5. And after 5 minutes there, after ingesting the glucose, their blood glucose levels have hardly changed. So they haven't got any of that extra substrate that's arrived. But if you wait a half an hour, as you can see, they peak at around 8 millimoles. So now you've got all this additional substrate that can come in as glucose 6-phosphate and be used through glycolysis. So physiologically, it would make much more sense to wait 30 minutes. Well, there's a mess up in that one, but that's okay. So we thought, okay, that's a problem with the Anderson trial. Maybe 40 minutes is much better to undergo the wait period for ingestion. But there are also some problems with this, and I'm sorry about the animations on this. They've gone off a bit early, but we'll get through that. So as I was saying earlier, this was the 2003 trial where they were cycling at 50 watts. Now it says in the research paper that the exercise intensity was done at a constant exercise intensity from start to finish. It does mention in there that the placebo trials had to be reduced in their intensity because the individuals couldn't keep at that intensity. But they mentioned nothing of the sucrose trials turning down the intensity. But as you can see with the orange circle, both the placebo and the sucrose trials were turned down in intensity within the first 3 minutes of exercise. Now, if you turn down the intensity, and it's actually quite an extreme turn down in intensity because if they're cycling at 50 watts, that first marker on the bottom left of the circle is about 25 watts. So they've actually got a 50% reduction in what they have said in the paper that they're actually cycling at, that you're going to see a very different heart rate response to the one they presented. And what I've loosely tried to draw there in the green is probably more likely a predicted heart rate response if they hadn't turned down the intensity during the early stages of exercise. And so while this shows that perfect magic bullet, oh, symptoms are all gone, I think it's quite flawed in the methodology because you can't turn down the exercise intensity and not expect the heart rate to be lowered with that as well. So what we're doing, this is the third study of my PhD, is we are trialing the Vissing trials, or similarly, we're doing 5 minutes before exercise with sucrose, 37 grams, versus 25 minutes before exercise. And we've picked 25 minutes before exercise because we believe blood glucose is going to peak about 30 minutes. So we don't want to miss that peak. But also it's in the first couple of minutes that individuals start to really, individuals with McArdle disease really start to display increased RPE and heart rate. So we wanted to hit right in that sweet spot. So that's why we chose the 25 minutes. So we had exercise control 24 hours before the trial, so no high intensity exercise. We also had 2 weeks of pre-trial cycling beforehand because traditionally this population is low, has a low exercise tolerance, a low adherence to exercise. So we wanted to make sure that there was no adverse effects to the trials. And then we also had dietary control, which is really important, I think, and wasn't done in either of the 2 previous studies. So in the dinner before the trial, we would give them a carbohydrate meal of 2.5 grams per kilogram body mass, which is really important because the only source of carbohydrate that individuals with McArdle disease can get is from their liver glycogen or exogenous sources that we would feed them. And if you don't have that standardized at the start of every trial, obviously that's a huge variable that we need to control for. And so we're controlling for that as well. So then they would arrive on the morning of a trial at 7 o'clock in the morning. We would cannulate, take a resting blood sample, and then they would either get a sucrose drink with 37 grams of sucrose, 25 minutes before exercise, a placebo, 25 minutes before exercise, and then in other trials, they would get a 37 gram sucrose 5 minutes before exercise or a placebo. Then what we would do is we would get them to cycle for 20 minutes. And the cycling at 20 minutes was at a constant workload. So previously we would get them in and we would get them to do a peak power output test. So we would then get them to cycle in these sessions at 60% of their sustained peak power output. And that was constant. We didn't turn it up or turn it down. And the way we got around having to turn up or turn it down is if individuals needed to stop at the trial at any point, we would measure how long those stops were for and how many that they would have. Because when you're exercising with McArdle's disease and you have an RPE of 15 or 16, you don't keep going, you stop. That's naturally what happens in the real world. But in research settings, they generally push themselves a little bit harder. So we wanted to give them that realistic real world setting of, okay, you would stop. And so we are now through 3 participants and we're on to 4 and 5 at the minute. And then our results at the minute are looking pretty solid for our hypotheses, in that we've got time again on the x-axis, and this is just the first 10 minutes of exercise. We're actually doing it over 20, but the real interesting stuff is in the first 10. And then we've got heart rate. And we're also measuring heart rate and RPE. We're also taking, obviously, bloods in there, as I said, so we can measure free fatty acids, blood glucose levels, lactate, and hopefully a few other things if we've got enough funding to do so. But as you can see from our first few participants, sucrose 25 minutes before exercise is the clear squares. And you can see that that has the lowest peak in heart rate, and it's also corresponding with the lowest RPEs, which we would expect because they would have the highest blood glucose levels before starting exercise. Interestingly, the sucrose 5 minutes before exercise still has an effect, but nowhere near as big. So we would suggest this goes against current guidelines. While we believe that sucrose obviously wouldn't have arrived at the working muscle, most of it wouldn't have, some of it potentially could have. There are neural effects of sucrose just in the mouth and neural receptors that allow for increases in exercise intensity and decreasing ratings of perceived exertion. And then you can see our placebo trials, which again mirror the placebo trials of previous research, where you're up around that 160 beats per minute. So we would say, yes, sucrose does work. However, it needs to be at a time ingested beforehand that's appropriate for the sucrose to enter the bloodstream and then enter the muscles at the points when it actually is required. However, we're still seeing with the sucrose at 25 minutes, it isn't this perfect line that goes up and then stops. There is still a peak in heart rate that then comes back down. So it isn't a magic bullet response, or we don't find it to be that so far. And then what we're also showing, the red P1, P3 is where our participants first stopped in their placebo trials. So you're actually seeing that after 2 minutes, after a minute and a half, participant 3 couldn't cycle any longer. So they actually had to stop on their placebo, and participant 1 had to stop after about 5 minutes. They were then able to continue to exercise, but it shows a big difference in actually having sucrose before exercise or not having exercise. So we've got another 3 participants to get through, hopefully within the next couple of months. That'll wrap up my PhD, which would be great. So watch this space for McArdle's disease research, I guess. Thanks for your time. And if you had any questions, I always love talking about it, so feel free. Yeah, I might have gone a bit over there, sorry.
No, we started with.
Thanks, Sam. That was really accessible for someone with a non-medical background. So thank you for your really excellent presentation. I think the results have really pragmatic, real-world relevance. So that's great to see. I guess I'm interested to hear what you would be saying after the research that you've done to people with disease, including your sister. Like, what ideally would you like to see in terms of how those guidelines would change? Do you think it is just a matter of number of minutes, or are there other things that need to change?
I think there's a lot that needs to change. So if we go back even to the guidelines that I did have here, that's pretty much the extent of it. And what I would say about though is the guidelines are predominantly have been enforced or developed by IMGSD, and IMGSD is a not-for-profit. It's a charity run by people with McArdle's disease. So they're not scientists themselves, but they do have some scientists that are helping them. And so as part of my second study, Rob and I have actually written out guidelines, that's really practical guidelines. This is the type of sugar that you need to have beforehand. This is the time that you need to have it. You need to have it all in one hit, and get them to understand the reason why you're doing this, that it's hyperglycemia, and that's where your energy is coming from. I do also think that it's not a practical measure for daily physical activity, because 37 grams of sucrose is still a lot of energy intake. Yeah, and so if you're doing that every time you've got to go for a coffee with your friends, you're going to have severe weight gain. And so I actually don't think it's a practical measure for daily activities, but what I really do think it is useful for is a training aid for exercise. And previously, when McArdle disease was first discovered, exercise was very shunned. Don't do it. It's very dangerous for you guys. But now we're finding that the more aerobically fit an individual can get, the more fat you can burn at any given intensity, exercise intensity. And because individuals can't use their glycogen, the more fat they can use, the less their symptoms become. So what we can use this for is to get them to increase exercise intensity so that we can increase their adaptation to fatty acid oxidation. And over time, we build their aerobic fitness so that their symptoms should reduce during any physical activity because they can burn higher levels of fat. Cool?
Sorry, I'll just get the computer changed over while.
Yeah, I'll jump out of the way then, yeah.
So that's very interesting with the sugar, like you were mentioning the fatty acids. And so have you tried mixing the sucrose with any ketone bodies or?
We haven't personally, but there has been stuff, probably the major thing going around with McArdle disease is ketogenic diets. Now, ketogenic diets I do see as having a benefit, and probably more for those maybe daily physical activities. However, you're still having the problem of anytime there is an abrupt increase in exercise intensity or physical activity intensity. So again, just walking up the stairs, ketone bodies still have to be oxidized as similar to fatty acids. And so you don't get that flush through glycolysis that's allowed for anaerobic activity. And so, yes, for slower activities, for lower grade activities, it would help. There have been some studies done with ingestion of ketone bodies. However, ingestion of ketone bodies can cause an insulin spike, which crashes free fatty acids, so it's a balancing act. There were, again, a few flaws in the methodology of the study, so it makes it very hard to find. They have also done infusion studies with free fatty acids, which have shown some benefit as well. But again, infusion doesn't really help for everyday living because it's not a practical measure. So I think there's still a lot of work to be done in this space. Yeah, it's such a rarely researched field, so it's still a lot of work to be done in there. Yeah, but good point, man.
We might leave it there, but we'll be having for drinks later, so you can continue with nothing. Thanks so much.
Cool.
Thank you. Okay, and second up, we have Associate Professor Rob Robergs. I have a very personal connection to Rob. I used to coach his daughter in gymnastics back in America, and the reason I'm in Australia is because Rob was my honors supervisor and encouraged me to apply to come over here. So very excited that I then met with Marek, who knew Rob, and this has all happened. Rob completed his PhD in Human Bioenergetics program at Ball State University. He then commenced a tenure track teaching and research job at the University of New Mexico, Go Lobos, and later returned to Australia working at Western Sydney University, Charles Sturt University, and commenced his current position at QUT in 2016. Rob has published more than 150 research manuscripts in peer-reviewed journals and authored 5 textbooks on exercise physiology. He is currently working on numerous research projects and manuscripts connected to the biochemistry of metabolic acidosis, exercise physiology, performance, and fatigue, modeling motor unit recruitment and its influence to muscle metabolism, improved equipment and data processing for oxygen uptake, VO2 kinetics, and maximal oxygen uptake, VO2 max, and lessons to learn about science from the history of science and scientific philosophy. Oh, that's a mouthful. We all have a lot to learn from Rob. Please come up and give us your wonderful presentation.
Okay, yeah, so hi everyone. So I've got to talk a bit about chemistry, sorry. I'm I'm an exercise physiologist, had a passion about physiology right from the get-go. I was an athlete as a younger dude. And then when I found that there's a scientific way to explore exercise performance, and it was steeped in chemistry, even though I was educated in a time when I didn't learn about chemistry much at all, I realized that was the stuff to learn. So I kind of did it mostly on my own. But when I developed the talk, you know, I've been commenting on and writing about and researching the biochemistry of acidosis now for almost 30 years. And, you know, it's been steeped in challenging the whole concept and construct of lactic acidosis. And I could talk about organic chemistry and biochemistry for hours. But I think the really interesting thing about my experience in this journey has not so much been about the chemistry and the biochemistry, but it's been about the personal story. So the story behind the story about what led to the challenge, what's the evidence, what are the experiences in life with the people you meet that end up pushing you indirectly and passively along a different road so that you are doing things that you never really planned that you wanted to do, and the challenges that confront you as a scientist and an educator about searching for the truth, right? And as a university academic, there's nothing more important than to teach the truth, right? If you're trying to educate a student, there's nothing more important than conveying knowledge that you, as best as you can possibly understand, is actually correct. And when I watched the start of the movie The Big Short, right, about the global financial crisis, I was first introduced to this quote by Mark Twain, which most American philosophers would really hate the fact that Mark Twain is the most noted philosopher in the US. But nevertheless, you know, it ain't, you know, what you don't know that gets you in trouble, you know, is what you know for sure that just ain't so. While at that time it had direct relevance to the global financial crisis, I instantly realized that it has direct application to anybody who wants to label themselves as a scientist, because if we're not taught to understand our own limitations and fear to a high degree what we're doing might be wrong, then we're not really able to identify if and when we are doing it wrong, right? You've got to have your eyes open and realize that along the way. So yes, I'll be talking about chemical structures a little bit, but I'm not going to focus on it. I'm going to talk about the pathways, and the multiple pathways that led me on this journey. And I think that's the really interesting stuff, and where it leads me now to understand in the bigger picture, based upon all my reading and thinking about what science is and how we should best pursue it. And if we understand that, then that means we're more directed in understanding how we best should teach, right, our students how to be better scientists. So along the way, I'm just going to try and bite my tongue and stop myself talking a lot about the chemistry, and we're going to focus on the bigger picture, because that's where I want to go. So just like Sam, I've got to click on it and then go, yeah, good. So my purpose, right, threefold. Certainly, we'll talk about the evidence that I've pursued to understand the chemistry. From that, because there was a time, as I'll talk about soon, when I was blasted and criticized and had to deal with a lot of antagonistic replies and responses and letters to the editor and replies and blah, 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 as a young academic, right? Because I actually started challenging lactic acidosis 5 years outside my PhD, right? And I'll talk about that journey coming up. And then that leads to understanding what science is and how to best pursue it. And then, right, making really you all realize that, right, gosh, we need to question everything, right? And we'll talk about why. And right, the last half of my talk is all about, right, the reasons why, right, we need to be very questioning about what it is we're trying to learn. Now, early in my career, this was when I was at Wake Forest University doing my master's degree in exercise science and cardiac rehab. Great experience, right, to be there in my 20s, working alongside cardiologists and pulmonologists and endocrinologists, dealing with using exercise to diagnose, right, and treat individuals with cardiovascular and pulmonary diseases and metabolic diseases, obviously with diabetes being the main one. Great introduction to what we're now trying to do in Australia, right, which is interprofessional practice. But back then in the States, doing it in the 80s without it being called interprofessional practice, it's the way, right, it was done. And thanks to the clinicians, they were very, very open to bringing in exercise physiologists, right, to understand the role of exercise in, right, the clinical practice, which was wonderful. But early on, right, so before, I studied organic chemistry before I went to the US. So I did that on my own at the University of Western Australia before I got accepted in the States, and I studied pure physiology because I knew as an exercise science trained person, I had poor background in that stuff, right? And even though I hated organic chemistry, it's probably the most important subject I've ever studied because it's had the most profound importance to how I function, right, through my career as a metabolic biochemistry focused, right, physiologist. But back in those days, I was intrigued to realize that, wow, there was actual dialogue in published journals that actually questioned how science was functioning. And Victor Catch, he's one of the authors of one of the main exercise physiology textbooks that I actually studied from as a younger student, was questioning, right, aspects about science and really challenging exercise scientists to be more critical in their inquiry and to challenge each other in the development of better theories. And even though I didn't know it, I was too naive then to really understand this. It was a prominent seed that was planted that became really, really relevant later in my career. Right, so this was during my master's degree, moved on to my PhD, where I studied more advanced biochemistry and bioenergetics and biophysics and really understanding the nitty-gritty of, right, energy transfer at the physical and chemical levels inside, right, living cells and tissues. And, my Leninger was my Bible. I would just read it for fun. Sounds pretty weird, but I realized it was totally important. And the more I delved into it, at that time, right, so I was still in my PhD days, we were taught that, right, certainly this was the lactate dehydrogenase reaction, and certainly it's an oxidation reduction reaction. And when you're involved in NADH and hydrogen ions and NAD, I would ask my biochemistry lecturer, I said, well, do these hydrogen ions that are involved in oxidation reduction reactions have any direct relevance to the cellular function? And the standard answer I would always get was no, they're just there to balance out hydrogen ions and ignore their presence. They have no direct role in anything with regard to cell function. And certainly if you went on, so back in the 90s, Peter Stewart put forward his physicochemical theory about causes for changes in acid-base balance, and it's very clinically relevant because a lot of clinicians abide by this as well as the traditional acid-base approach. But there's a prominent quote there in his 1983 publication that was really concerning to me about the assumption was that, again, like the biochemistry lecturers were telling me as a student, that just don't worry about the hydrogen ions that are involved in chemical reactions. They have no relevance. They can't directly change anything. And then Peter Stewart had that as his fundamental base assumption, so that it would be that, well, everything else, something else must be causing, right, these changes in cellular pH. But even when this was presented in Leninger, showing the ionic forms of what would chemically be labeled as, right, carboxylic acids, right, so it's a carboxylic acid functional group. Then there's quotes in the biochemistry textbooks that, well, it's the production of lactic acid, right, that's causing acidosis. So to me, that was an anomaly. And there are many anomalies that challenge the lactic acidosis construct that date back to the 1970s. Well, I cottoned under that really, really early, but I didn't do anything about it because I was a student, right? I must be wrong. I must be thinking incorrectly. I've got to listen to my instructors. And so I graduated, right, did my UNM stuff. And then I got to talk to a biochemist in biology, and at the University of New Mexico, the biology department was 90% plant biology, right? So there are botanists, no human biology, no vertebrate biology really. And he pulled me aside one day and said, Rob, what's the deal with your field? You just keep talking about lactic acid, and there's absolutely no such thing as lactic acid in any cell. And that was the first time I'd ever heard that. So there's a comment done in a very nice way. It wasn't a really abusive, insulting way, that made me think, right? And so I realized, gosh, you know, I've been taught all this about lactic acid, and then here's a established biological scientist telling me that there's no such thing. And so I just got to work, right? And so I was really concerned about my whole career being invested in a field that was on the wrong paradigm, right? And I wanted to know the truth. And so after my second textbook, so I wrote my first textbook about 5 years in after my PhD, and then followed by 2 or 3 other ones. And then the editor of the Journal of the American Physiology Journal, or the American Journal of Physiology, emailed me and said, Rob, I've read your latest book, saw the biochemistry section, you need to write a review on, right, your chemical rationale for challenging the lactic acidosis construct. So that was my 2004, right, manuscript. And I'd written stuff beforehand, but, you know, I wasn't really prepared for the critical confrontation that was unleashed at that time. It's ingrained in clinical science, it's ingrained in biological science across so many subdisciplines that relate to biology and chemistry. And then, of course, I didn't know at that time, but essentially builds to mean that, well, I'm not just challenging lactic acidosis, I'm challenging the fact that generally the concept means that no cell can produce a metabolic acid, right? So it spans, right, biochemistry and chemistry, as well as all the applications of that through clinical and applied science. And so it's a big issue. And yeah, I just put the timeline on here because part of this manuscript was to explore the history of lactic acid, and the history explains a lot. And the latest work, or the most recent work on acidosis, is really quite strange. I've done quite a lot of it, but I would say in the last 15 to 20 years, the acid-base chemistry and acid-base physiology has been in a state of turmoil because we can't rest our hat on the lactic acidosis construct. Then what is there to explain acidosis? And that then made me question about science and how does this work, and is this usual or unusual, right, about science, about things being right versus wrong, and if things are wrong, wow, what does that mean? So part of the criticism that came to me from the 2004 manuscript was that, well, Rob, you're just counting hydrogen ions, you can't, they're pH dependent, even if there are covalent modifications in the use or so consumption or production of hydrogen ions, there is still pH dependent ionization going on, or association, depending upon what the dissociation constants are. So I had to school myself in a re-understanding of not just association constants, but competitive cation interactions with negative charges, because hydrogen ion isn't the only, right, positive ion that can do all this. And then I had to conceive better understandings of how then we can quantify total hydrogen ion exchange through chemical reactions. And so I talked with a computational chemist at the University of New Mexico, and he introduced me to this alpha equation. And sorry, it gets much lighter after this. So I had to do alpha equations across hydrogen ion involvement, calcium, magnesium, potassium, sodium, across all the known dissociation constants for all the known metabolites of the reactions of cytosolic energy metabolism. Did it all on my own, thousands and thousands of reactions. So I had to really improve on my math in all of this, but I did all that stuff, and that led to this 2017 publication, and that led to a review, well, a modeling approach of then applying this to exercise. And essentially what this means is that, well, if you look at the hydrogen ion, right, exchange, right, the lactate dehydrogenase reaction actually has hydrogen ion consumption, right? When a cell produces lactate, right, it's almost an equivalent one hydrogen ion, regardless of the pH of the cell, because it's somewhat pH independent. So the more lactate a cell produces from a hydrogen ion perspective, the more hydrogen ion consuming it is. So if there's acidosis, it must come from somewhere else. The pyruvate kinase reaction is also proton consuming. The most proton releasing reaction from glycolysis is actually the glyceraldehyde 3-phosphate dehydrogenase reaction. And then the ATPase reaction is inherently protein releasing, although that decreases dramatically as pH goes down, but that's getting much more complex. So my 2017 manuscript, I was able to devise all these pH-dependent hydrogen ion exchanges for all the main chemical reactions of interest in the cell. Sam talked about glycolysis, and so this was really for all of those. And right here you see the glyceraldehyde 3-phosphate dehydrogenase reaction. Very simple reason that explains it is just because as pH goes down, inorganic phosphate, which is accumulating during intense exercise, actually gets another hydrogen ion. It goes to dihydrogen phosphate. So that means in this reaction, there's now an added hydrogen ion being released because the inorganic phosphate from the cell in solution is being used as a substrate. So there's totally explainable biochemical reasons for why that's the case. And that led to the modeling. That led to us then working with Marek and Steve Curry from the toxicology department at Bendigo University in Phoenix, Arizona. Wanted to work on this manuscript for, gosh, this Marek maybe 8 to 10 years, but Sam came along, kind of stirred up me a bit and got me off my chair and kind of said, Rob, get back to it. And so, right, help Sam have that be the study. So we published this earlier this year, right, as part of his PhD dissertation, and Marek and Steve helped out with content and editing as well, obviously. And we kind of sorted out, well, here's the more complex hydrogen ion exchange during diabetic ketoacidosis, or really just any ketoacidosis, right? And we showed that ketone bodies aren't produced as acids, just the same as lactate isn't produced as lactic acid. And there are other reasons for the hydrogen ion exchange. And so, yes, that was quite relieving. And again, it kind of leads to, if there's a reaction that involves water, so hydrolysis, you're going to have hydrogen ion involvement. And like I mentioned, also oxidation reduction reactions. And so, but some of the criticisms I've had early on was that, well, Robbie is talking theoretical stuff. It's fine. It's evidence. Yes. It's science. Yes. But we need to see some hard data. So when would this have been now? Back in about 2006, I was working with another of my PhD students, and I was devising a PhD research study for her. She was an Iranian student, but unfortunately, she got forced to go back to Iran, right, in a pre-arranged marriage, and that ended that PhD. And I couldn't finish that work, but what I was devising was that, well, there's got to be a way to directly identify this. So I developed essentially cocktails for the different reactions, right? So an in vitro system, and I was doing the major reactions, and this was going to be part of her PhD because this is really needed to solidify the evidence to show that, yes, hydrogen ion involvement in chemical reactions can directly influence biological pH. And so just for example, for this lactate dehydrogenase reaction, this chemical depiction would tell you that if you produce lactate in a cell, pH should go up, right? So I just did that, right, in an in vitro system, and lo and behold, if you run this reaction inside, right, a flask, right, or a test tube, had a pH meter involved, lo and behold, right, pH goes up. Did the same thing for ATPase, right? pH goes down, right? And I did that for several reactions with glycolysis. That was the whole point. And this has been something I've been wanting to do for, gosh, almost now 14 years. And boy, if Marek's plan comes into fruition, right, we could be off to the Czech Republic and doing this work over there right next year. Because it's totally needed, right? And it's not just going to challenge exercise spheres, but it just challenges the whole, right, fields of organic and biochemistry to reveal that, wow, right, hydrogen ion involvement is biologically relevant in chemical reactions, both as substrates and or products. So what is most interesting about all of that? That 10 more minutes, or you want me to?
We normally finish at 5, but I'm sure if anyone needs to go, that's all good. But
We're happy to stay.
Not to blame Sam, but I started late. Yeah,
We started.
we started late. All right. So what's most interesting? And I can brush through this. Well, to me, it isn't the chemistry, right, or the biochemistry, right? It's Well, if you accept the fact that the data is the data and the evidence is the evidence, that means there are mistakes that have been made. And so why have we got it so wrong for so long is the prominent question that I'm not the only one that asks this. Other people in audiences that I've talked to have asked this for many, many years now. And I've been on this more recent journey for the last 12 years, but trying to understand what is it about science that enables things or mistakes to be made. And so why are there errors in science? And the fact is, the main lesson I've learned is that there have always been errors in science. There will always be errors in science because fundamentally speaking, we just don't know what we don't know. And we're all taught from prior decades, sometimes longer, of knowledge. And unless we're really wary of potential errors in that knowledge, we're never going to find those errors because we're not looking for them. And my first readings were from obviously Karl Popper. And this quote really made sense to me, right? Because Karl mentioned, I think quite brilliantly, that unless we're aware that part of our education, although it enlightens us, it can also confine us if we're not really, really careful in how we use that knowledge. If we don't use that knowledge to improve our knowledge, right, because we just accept what it is that we're told, we're not really being scientific, right? And we're not being well educated. And we can expand that into, well, that means we need to educate our students to be more challenging, more critical, and we do, but I don't think we do a very good job of that in universities. So that was intriguing, right? That made me think about, wow, okay, fair enough, right? We're not always taught correctly, right? It's our responsibility to use what knowledge we've gained to make sure that we understand things in more detail. So that meant that, yeah, certainly my background wasn't necessarily the best, even though I received this phenomenal education right in the US. And that led me to Kuhn, and most people would say that in the world of science, Kuhn's really, really important. If you want to understand science, you've got to read Kuhn, right? The Structures of Scientific Revolutions. Anybody here read this book? No, some of it. I think every teacher, right, every scientist needs to read Kuhn, but not from the context that what he's saying is always right. There's a lot I think Kuhn doesn't get right. But the fundamental features of Kuhn, and we don't have time to go into this in a lot of detail, but he identified what a paradigm is, and you can just argue that as an interpretation, a theory, an understanding, a method that we kind of accept, right? And over the history of the physical sciences, which Kuhn wrote his book on, right, Kuhn's observation was that generally, if there's a paradigm that's been accepted, the fundamental mainstream way that people behave is to just accept it and not challenge it, okay? And he labeled the practice of science to be based upon using an accepted paradigm to try and solve problems, but not necessarily challenge it. And he called that normal science. Totally annoyed Karl Popper. He didn't like how you could call anything normal that wasn't challenging. But more about that in a little bit. And then when there's evidence that challenges a paradigm, it is also normal for most scientists to disregard it. They'd rather stay mainstream and accept the prevailing paradigm. Generally, anomalies build over time. There's not one person necessarily that provides the ultimate new change to a paradigm. And Kuhn's examples were about oxygen and electricity, and others, right, prominent in the physical sciences of the time. And he would label scientists who could ignore the paradigm and step outside and see things differently as pursuing extraordinary science. Crisis is formed, right, and that might lead to a paradigm shift. And, right, Kuhn's argument and has been read and interpreted. I mean, well, that can also apply to other disciplines besides the physical sciences. Right? However, what most people don't realize is that Kuhn also clearly identified that his view of normal science was that it was a really flawed enterprise, right? And he described negativities about normal science that to this day, I don't think many people who have read Kuhn would even comment on, because when I read commentaries about Kuhn, they've just praised it quite remarkably, but Kuhn was really quite self-critical, right? And none of these traits of his perception of normal science are very flattering, right, about science at all. And unfortunately, my perception of my experience in challenging a main paradigm is that there's so much inertia, right, and driven force to regain or sustain an acceptance of a pre-established accepted paradigm that they're really hard to change. And so you can draw this up, right, in a diagram, if you like. We've got normal science, problem solving. It just stays within its accepted paradigm, if you like. And then you have to kind of step outside of that. You've got to have some capacity to see things differently, able to see the anomalies that are happening, and then to fuel the change. And these changes are occurring all the time in disciplines, right? It's just a slow process, unfortunately, because of this dominance, right, of normal science. Right, and probably didn't like it, right? You know, why call it normal if it's so dysfunctional? And if you are more diagrammatically, right, focused, right, it'd be like saying we're taught to follow a paradigm, follow a road, and if we stay within that focus, right, we're unable to see all the anomalies that are out there, which would lead us to the solution, but we're off doing something else, right? And that has been common and is common, right, to disciplines throughout the history of science, and changes eventually happen, but it takes a lot of change based upon a major crisis, right, to occur. So certainly I think we need a paradigm shift in how we both understand science and teach science so that our students can really comprehend that, wow, there's a major responsibility in pursuing correct scientific practice. And what I did, I'm finishing a manuscript right now with Sam and some other of my PhD students. And this is just part of an endpoint to our last manuscript of a 3-part series. But these are just all tips about, right, how to teach our students and to be aware ourselves about how to function so that we improve the quality of the science that we're pursuing, and being aware that, wow, we are somewhat limited if we're not challenging, right, what it is we know. And there's always more to learn, and that can only improve how we pursue science. So there's a whole list of stuff. We're running out of time. There's actually quite a bit. There's like 3 pages of it. But in this manuscript, right, it all ends in, well, if you want to improve the science, right, this is what I think, right, you need to do. And it's about interacting with colleagues, publishing. If you don't like your peer review, complain, right? If you've got evidence base to complain, do it. Challenge your journals, right? Challenge your editors of journals. And unless we do that, then nothing's going to change. So that's a synopsis. That's my talk. And I hope you understood my journey. Thanks.
One question and a few questions.
Can I ask a really naive question?
Yeah.
So a lot of people really enjoy a fantastic sort of journey. I go to the gym most mornings, right?
Yep.
My trainer turns around to me and says, "Oh, you've got to shake that lactic acid out."
Yep.
What is it I'm actually shaking out? What is it that is actually causing that?
Well,
sort of crank response?
So one, because of the acceptance of lactic acid,
Yeah.
right? Not a lot of research to directly answer the question, but there's a lot of research that can be tapped into that does provide some answers. So contractile failure, and we've known this now for 35, 40, 50 years, right, is more likely largely caused by the role of inorganic phosphate in interacting with calcium and impairing calcium availability for the whole contractile mechanism, right, to work. The shakeout concept is probably more about the increasing edema in and around muscle because of the accumulating metabolites that are occurring from the more intense exercise. So they need to be washed out, right? And that's just a vascular process that's going to happen. So yeah, there are other research proven mechanisms that are better able to explain, right, those aspects of muscle dysfunction and the feeling related to that than, yeah, the whole concept of lactic acid.
So it's
Yeah.
much more complex than just.
Oh, it is, yeah,
Okay.
yeah. And then there's one section of one of the manuscripts we've written. I mean, if you if you think about all the acids that are in the body, right, nucleic acids, deoxyribonucleic acids, right, the keto body potentially has acids, all the other acid metabolites of glycolysis, right? You know, lactic acid's just one. So that, I mean, there's so many grounds to question why we've blamed one molecule when there are so many other acid, right, chemically labeled molecules in cells. It's very quite strange. Good, all right.
Okay, we'll just close off quickly. That's all right, I can get that down. So thank you very much, Rob and Sam, for joining us here in Canberra. It's been a delight to have you guys here. Next week is our final week for our SMP seminar for 2023. We have an exciting seminar to celebrate Transgender Awareness Week with collaborators from SMP, Canberra Health Services, and A Gender Agenda. We will also be hosting a post-seminar picnic from 1 to 2 p.m. So please make sure you register if you haven't already done so that we know how much to cater for. We look forward to seeing you there or online. And I think we're good to go. We'll wrap it up there. Thanks, everybody.